Hi, in this video I will talk about bilirubin metabolism. So the precursor for bilirubin metabolism is heme. So bilirubin is formed from heme. So you must be familiar with the term bilirubin because bilirubin level increase during jaundice and that causes a yellow pigmentation in the skin. So we need to talk about bilirubin metabolism. First, we would look where the heme is produced. So heme is produced during erythropoiesis in the mitochondria. So once heme is produced and hemoglobin is formed, RBC would be circulating all over the body. And you can see RBC is circulating all over the body. But RBC have a lifespan of 120 days. After 120 days, when RBCs grow old, they would reach the spleen. So spleen is the graveyard of RBC. So in the spleen, there are splenic macrophages. So these macrophages would take up those old RBCs and they would actually degrade the RBC. So inside the RBC, we have hemoglobin. So hemoglobin would be degraded into two parts. One is heme, another is globin. So heme would be further broken down and ferrous would be, that means iron, would be recycled and used further, and heme would be broken down and the other cyclic compound of the heme would be broken down and it would form bilirubin, and globin would be broken down and recycled. So first what happens, this is the structure of heme. You can see it has 4 protoporphyrin ring, and these 4 in the center of this 4 ring there is a ferrous which is chelated from the lone pairs of nitrogen. So this heme would be broken down by the action of the enzyme heme oxygenase. This heme oxygenase enzyme would break down this heme to form biliverdin. So biliverdin is an intermediate, and after that biliverdin reductase would actually convert biliverdin into bilirubin, and in this process NADPH is converted into NADP. So this is a reduction step and the reduction potential comes from the NADPH. So you can see in the biliverdin and in the bilirubin, inside the red box you can see more hydrogen is added. So this is how bilirubin is formed. This bilirubin which is formed by reduction of biliverdin is actually unconjugated. Unconjugated bilirubin is insoluble. So unconjugated bilirubin would move to the circulation, and so it, as it is water insoluble, it have to work with any carrier protein. In this condition the carrier protein would be albumin. Albumin would conjugate with it and it would carry the unconjugated bilirubin to liver, where it would be further metabolized. So in the liver, unconjugated bilirubin would be converted into conjugated bilirubin with the action of UDP glucuronide transferase. Now unconjugated bilirubin was insoluble, but UDP glucuronide transferase would add a glucuronyl group which would make it soluble, and this soluble bilirubin would be secreted into the intestine, and inside the intestine it would pass through the intestine and ultimately it would reach the large intestine. So in the large intestine further steps take place. So in the large intestine, so you can see in the bigger picture the conjugated bilirubin is converted into urobilinogen upon bacterial activity. Then this urobilinogen is further converted to stercobilin, and stercobilin is excreted in with the fecal matter. So you can see some amount of urobilinogen is converted into stercobilin, and some amount of urobilinogen is converted is absorbed and it moves to kidney, and inside the kidney some amount of urobilinogen is converted to urobilin, and urobilin gives the urine its characteristic yellow color, and the other portion of the stercobilin is actually excreted and excreted into the stool. So that is why our fecal matter or stool has a characteristic brown color. So this is how bilirubin is actually eliminated from the body. If bilirubin is not eliminated properly, then there is accumulation of bilirubin, which is common in jaundice. That is why we need to study the bilirubin metabolism. I hope you enjoyed the video. If you like my video, please like it, subscribe to my channel, and don't forget to leave your comments below. Thank you.